Bismillahirrahmanirrahim. Myoglobin can bind only one molecule of oxygen because it contains one heme group. In contrast, hemoglobin can bind four molecules of oxygen at each of its four heme groups. The degree of saturation of these oxygen binding sites on all myoglobin or hemoglobin molecules can vary between 0 when all sites are empty to 100% when all sites may be full. Pulse oximetry is a non-invasive indirect method of measuring oxygen saturation of arterial blood based on the differences in light absorption by oxyhemoglobin and deoxyhemoglobin. Myoglobin is a heme protein present in heart and skeletal muscle, functions both as oxygen reservoir and oxygen carrier. That increases the rate of oxygen transport within muscle cells. Myoglobin consists of single polypeptide chain that is structurally similar to individual polypeptide chains on the tetrameric hemoglobin molecule. Myoglobin is a compact molecule with 80% of its polypeptide chains folded in alpha helix, as shown here. These alpha helical regions are labeled A to H. The heme group sits in a crevice lined with non-polar amino acids with exception of the two histidine residues. Proximal histidine binds directly with ferrous of heme. The distal histidine does not interact directly with heme but helps stabilize binding of oxygen to ferrous. This creates a special microenvironment for the heme that permits reversible binding of one oxygen molecule. Hemoglobin is found exclusively in red blood cells, where its main function is to transport oxygen from the lungs to the capillaries of the tissues. Hemoglobin A, the major hemoglobin in adults, consists of four polypeptide chains held together by non-covalent interactions. Each chain or subunit has stretches of alpha helical structure and hydrophobic heme binding pocket similar to that of the myoglobin. However, it is more complex than myoglobin. For example, it can transport protons and carbon dioxide from tissues to lungs and can carry four molecules of oxygen from lungs to cells of the body. The deoxy form of hemoglobin is called the T or taut form. In T form, the two alpha beta dimers interact through a network of ionic bonds and hydrogen bonds that constrain the movement of the polypeptide chains. Taut form is low oxygen affinity form of hemoglobin. The binding of oxygen causes rupture of some of the polar bonds between the two dimers, allowing movement. Specifically, the binding of oxygen to heme iron pulls iron in the plane of the heme because iron is also linked to the proximal histidine, F, also labeled as F8. It is present on this alpha helix. The resulting movement of the globin chains alters the interface between alpha beta dimers. This leads to a structure called R or relaxed form. This is a high affinity form for oxygen. Here the structure of hemoglobin and myoglobin are shown side by side. Although less than 20% amino acids are identical between myoglobin and either of the alpha or beta subunits of the hemoglobin, but all the three polypeptides share the same protein fold called the globin fold, which contains 8 alpha helices. Two critical histidine residues are important for oxygen binding in globin proteins. One residue, the proximal histidine residue, coordinates with ferrous directly. Other residue, labeled as distal histidine residue, coordinates to bound oxygen, which coordinate to ferrous. In absence of oxygen, the ferrous is not in the plane of the porphyrin because the ion is too large and heme is puckered, as shown in the above diagram. However, upon oxygen binding, the shared electrons result in slightly smaller atomic radius for ferrous, allowing it to move into the plane of the porphyrin ring as shown below. So myoglobin has one polypeptide chain, hemoglobin has four polypeptide chains. Hemoglobin transports oxygen, whereas myoglobin, apart from transporting, it also serves as a reservoir for oxygen. And taut or deoxyform is a low affinity form for oxygen, whereas the relaxed form of hemoglobin has high affinity for oxygen.